So, hello everyone. Can't hear you. Can you hear me now? Hello? No? Yeah, it's better. You need a little. Okay. Can you guys hear me now? Okay. No? On your lapel. What about now? That's better. That's better? Okay. Yeah. Hello, everyone. Welcome to RMI Sensory Theater. Thank you, Kyria, for explaining what the theater is about and what we do here. So I'm going to be your host this afternoon. Welcome to our speakers event. We have a lot of diverse talks today. So we have talks about nematodes, which are these tiny creatures. We are going to be talking about yeast. We're going to be talking about bees and how to create your own bee heaven. We're even going to talk about dinosaurs today. So it's going to be a really amazing event. And yeah, can't wait to get started. So let's introduce our first speaker for the event. Our first speaker is a PhD candidate in plant pathology. So she comes from the tallest mountains in the world, but studies the tiniest animals in the world as well. So, and when she is not doing research or like not in the lab or in her computer, she likes crocheting and she likes to read and she also likes to swim. And turns out the first speaker is me. Okay, let's welcome me. Okay, so the first talk is again going to be about nematodes. So I'm going to be taking you to a wonderful world of nematodes today. Okay, just by raising our hand, who went to the nematode booth today? Well, there's so many people who went. Okay, that's pretty cool. So I don't have to explain it to you a lot what nematodes are, but I'm going to do it anyway because that's the favorite part of my job, right? So nematodes are actually the most abundant organisms on Earth. One of the famous nematologists, also called father of nematology in the US, once said, if everything in the world disappeared except nematodes, we would see a ghostly outline of mountains, lakes, and trees made of worms. So, because there are so many of them. So what are nematodes? Nematodes are actually unsegmented roundworms. So they're not segmented like earthworms. They're unsegmented, they have cylindrical body. The ones that you see here is actually an electron microscopy image of a very famous nematode called elegans. For those who went to the booth today, if you saw some of those crawling around in the computer, so those are the C. elegans, and this is what they look like very close up. So roughly 80% of all animals on Earth are nematodes, and they're found everywhere. They're found in the ocean, they're found in your backyard, beneath what we are standing right now, outside over there, or in the Antarctic ice, and even in movies. And majority of these nematodes are free living. So they are free living, they're feeding on bacteria, they're just chilling, living their lives, right? But some of them are parasitic as well. So let's talk about some of these nematodes today. But before that, we have been really influenced by nematodes in Hollywood. These are some of the famous Hollywood nematode worms. So the first one that you see, any guesses? This is a famous one. Yeah?
It's the sandworms of dude, Shai-Hulud.
Yes, that's Shai-Hulud from Shai-Hulud from Sandworm, that's right. And have you guys watched this movie called Tremors? It's a really old movie. Have you watched it? Yeah, it's a really old movie. And then there are these Mongolian death worms. So these are the worms that are one of the famous folklores in Mongolia as well. And the third one, I don't know if you guys heard about this. This is a series called The Strain. Have you heard of it? So in this series, nematodes turn people into vampires. It's a pretty cool show, and I really enjoyed binging it, actually. So nematodes-inspired fictions, they look really scary, right? But the thing is, these nematodes were inspired by free-living nematodes, which are actually really harmless nematodes. And they were inspired because of how they look anatomically. If you look at these horns, as in these horns as well, they look very similar, right? So these are actually free-living nematodes, not harmful nematodes. So let's talk about these good guy nematodes, the beneficial ones. These species of nematodes hunt garden pests like grubs or fungus gnats, and they kill them without the need for harsh chemicals. So what they do, these are called entomopathogenic nematodes. So what they do is that when they see an insect, like a grub, for example, they go inside this grub, and inside this grub, they release some kind of bacteria. And as they do that, the grub dies. And inside that grub, they start taking all the nutrients in and multiplying inside the insect. And once they deplete this insect, they get out of there again, and then they start moving on to find other insects or pests. So how do they kill these pests? So here you can see these grub, like a big one, right? And then around here are some nematodes just killing them or unaliving them. So what happens is that these nematodes harbor special bacteria in their guts, and these are actually the bacteria that are released inside the insect body. And these bacteria are the ones that produce toxins. And these are the toxins that are killing these insect hosts. So now we have a plant, right? A really happy looking plant. And now there are good nematodes there. So these good nematodes are now everywhere. Like they're happy. The plant is really happy, doing happy. But every hero needs a villain, right? So now there are these plant pirates that are going around trying to get nutrients from the plants and trying to get your garden plants, like really damage your garden plants. So these are called plant pirates, what I like to call, and this is what we study in our lab. So let's talk a bit about these plant pirates. So have you ever plucked, if you have a garden, have you ever plucked a plant and then the roots look like this? No? Okay. Yes, somebody said yes. So these are actually caused by plant parasitic nematodes, and then they parasitize every kind of plant there is. So for example, this is one of our field samples that we collected. So these are the tomatoes. Tomatoes that were infected by root knot nematodes. This was collected by one of the PhD students in our lab. The second one that we have are potato cyst nematodes. So these are the cysts that you see over here, and then you can also see sometimes in the potatoes itself. Third one, some plant parasitic nematodes are also in the leaves. So these brown stripes that you see are the symptoms produced by some of these foliar nematodes. Lots of nematodes also damage corn. So this is an example of a nematode damaging the corn roots. And then there are also garlic nematodes that basically parasitize garlic as well. So nematodes can parasitize like all kinds of plants. So how do they attack the plants? If you look at this video, this is a really cool video. And in this video, you can see something moving here, right? And it's moving here as it's going inside the plant cell. And this is one of their major weapons that they use. So this weapon is called stylet. So the stylet is like a needle or a syringe that moves in and out of the nematodes as they go into the plant cells. And this is how they get into the plant cells. This is how they release chemicals inside the plant that helps them parasitize the plant, and that helps them manipulate the plant as well. So how do I check if my plants have plant parasitic nematodes? There are a lot of things we can do to check. But the above ground symptoms that you usually see are sometimes look like nutrient deficiencies or something else. Like you can't see it just by looking the plant outside. What you need to do is you need to pull out the roots and look for like galls or lesions or stubs inside the root in the roots. And also another thing to do is to compare your healthy plant with the sick plant as well. And also maybe consider a lab diagnosis. For the lab diagnosis, there are multiple things you can do as we live in California. So you can go to your UC Cooperative Extension Office or contact your master gardener programs, which can help you like decide where to go or like if you even need to test your soil or test your plants or not. And if you do test your soils, make sure that you are giving the samples, like for example, not just your leaf or just the outside of the plant, but the entire root system and the soil as well, because that's important in order for the scientists to say whether it is infected by nematode or not. So these are some of the stuff that you can do to check if you have plant parasitic nematodes in your garden. There are some management strategies as well. The first thing to make sure is to start clean. So start with clean and healthy plants and clean tools beforehand. The second thing is to rotate your crops and use like resistant varieties. There are a lot of resistant varieties available in the market that you can use. And another one is removal of dead or infected plant material that can also then transfer nematodes to clean plant samples, clean plants as well. And then lastly, add compost and organic matter so that everything in the soil is healthy. And this is also one of the ways that you can avoid having nematode issues in your garden. So now we talked about the good nematodes, and we also talked about the plant pirates nematodes, right? What are we missing?
The morally gray ones.
Sorry?
The morally gray nematodes.
The what?
The morally gray.
The morally gray nematodes. Okay, like the in between. Yeah. Yeah, like C. elegans, right? But that's not what I'm going to talk about today. I'm going to talk about the ones that infect the humans and also our pets. So let's talk a little bit about these animal parasitic nematodes. So these animal parasitic nematodes can be roundworms, heartworms, or hookworms, and they can infect people too. And monthly preventives really help. So there are like medicines or things or yearly medicines that you can give your pets. So if you think about the heartworm is transferred by mosquitoes. When mosquitoes bites either a person or a dog, that's how these like filarial parasites, which are very teeny tiny, they go into the bloodstream and then they get to the herd and then they cause a lot of problems to your dogs. And again, when another mosquito bites this dog again, then those filarial parasites are again transferred to the mosquitoes. They go into the head of the mosquitoes and through the proboscis, like it again goes into the dogs or other dogs or also humans as well. So it's kind of like a continuous cycle. But there's always prevention. Prevention is the key, right? So preventing the mosquito bites or keeping your surroundings clean to avoid water buildups, to avoid the mosquito buildups, or year-round vet-prescribed monthly medication or yearly shots for the pets. Like these are the things that all the pet owners already do, and like this is actually really important so that your pets don't get heartworms. So now we talked about the good nematodes, the plant pirates, we talked about the animal parasitic nematodes, right? So let's zoom out a bit. And then think about our whole ecosystem. So in our ecosystem, these nematodes, they perform a lot of duties. So they are fungi eaters, they are bacterial feeders, they are predators, there are also root feeders that we talked about. But the thing to remember and the thing to take away from today, that I would like you to take away from today, is that nematodes are everywhere. Like when you stand on the ground outside and look down, like that's where you can, dozens of thousands of nematodes are right beneath your feet. And they're important for our ecosystem, even though they are bad, and there are good ones. But most of them are good. Like most abundant nematodes are good nematodes, and they're very important for our ecosystem. So this is one of the things that I want you to take away. And they're very sensitive to changes in our ecosystem. So they are the indicators of our soil health as well. And nematodes are basically quite helpers that help us keep the ecosystem running. With this, I would like to conclude my talk, and let me know if you have any questions. Plus, we do Biodiversity Day every year, so this is how we showcase our nematode collections every year in Biodiversity Day. It's pretty sad that this is going to be my last Biodiversity Day, which is kind of crazy because I've been doing it for 3 years. I missed last year. So this is going to be my last year of doing this day, and it's a really good day where we showcase all of our collections. And UC Davis has a really good collection of nematodes. So I hope, not me, but somebody else who's going to be standing there is going to see you next year as well. Thank you. Any questions?
I have one really short question while the next person sends them. And this makes my own paranoia. Should there be something I should do to prevent me getting hard ones?
Avoid mosquito bites is one thing. So it's usually it's filarial nematode. It's you get it with mosquitoes. So preventing mosquito bites, preventing any stale water around where there are mosquitoes. So that's how you can prevent it. And there are medicines as well. There are preventative medicines for deworming. Yes, yeah. All right, thank you. So I'm going to introduce Samantha. So our next speaker is Samantha Murray, and she is the Bee Haven Garden and Education Coordinator at UC Davis Bee Haven, next to Harry H. Laidlaw Jr. Honeybee Research Facility. She's passionate about pollinator conservation and garden care. And Samantha began her journey at UC Davis El Niño Bee Lab as an undergrad researcher studying how plant chemicals in diets affect honeybees. And today, she leads tours, educational programs, and outreach efforts that inspire appreciation and care for bees and other pollinators. So can you please give her a big welcome?
Thank you so much. Yeah, so can I have a show of hands of anyone who visited the Bee Haven Garden today? A couple? Okay, oh, well, awesome. If you haven't gotten the chance to visit today, which is most of you, just know that we are open from dawn to dusk. Oh, thank you, great. Every day and even weekends too, but if you want a more specialized tour, you can reach us at Bee Haven at ucdavis.edu. You can look at our website, and we do tours for classrooms, but individuals as well. So here's some of our pictures. This is our Miss Bee Haven. We've collaborated with the UC Davis Art Science Fusion program. So there's so many great art pieces in the garden, which help kind of show you that that's a beautiful honeybee, and you get to see it right up close. Okay, wrong way. Okay, so why is it so important for us to care about pollinators? Well, pollinators are facing habitat loss and feeding loss due to urbanization, and we are super dependent on our pollinators. 1 in 3 bites of food we eat are from a pollinated source. Bees also pollinate clover and alfalfa, which livestock eat. So bees cascade through our whole food system. So we do know how to help them, just planting pollinator-friendly plants. I'll get into that. And if we all do just a couple of plants in our own garden, it really does add up and make a difference. Monoculture is not enough nutritional value for our pollinators, so any additional forage we can provide for them makes a huge difference. Okay, so what are some bees that we have here? So this is the carpenter bee, and oftentimes people will confuse the carpenter bee and the bumblebee because they are big and kind of fuzzy looking. But the carpenter bee female will be black, And then this male carpenter bee is called the valley carpenter bee. And his nickname is like the teddy bear bee because it kind of looks like a teddy bear. Some other great bees we've got, digger bees, longhorn bee. You can see why because of that long antenna. And then we have more ground nesters. And so the leafcutter bee is ground nester as well as bumblebee, miter bees, and these mason bees will nest in these cavities. And so you can go home and plant your own native bee home. And I will talk a little bit about that later, but it's just cool to think about, get some visuals of what are the bees we're talking about here. So when bees are out and foraging, they're collecting nectar, pollen, resin, and oil. So nectar provides them carbohydrates, minerals, and micronutrients, but pollen is really rich in proteins, and they'll turn the pollen with their enzymes into what's called bee bread. So it's fed to the brood, the baby bees, and yeah, like I said, large-scale monocrops, they don't meet all the nutritional needs for bees. Resins, so for honeybees, they'll line their beehives where we've got multiple boxes we're working with. They'll seal that to let other pests come in. So they'll seal that with a type of resin, which they'll get from tree sap or sages as well. So that sages are great for bees. So to think about some concepts of creating your own pollinator haven, we want to think about providing year-round bloom for our pollinators, having something that's going to be in bloom each season of the year is going to allow them to have their resources. So if you're local to this area, we have our plant lists posted. We have our native plant lists, and everything that we have in our garden is on our Bee Haven website. You can also, research your local nursery and just really educate yourself on IDing plants and all the apps that can teach you what's local to you. Also, instead of scattering around like a great pollinator, you look up pollinator plants, but you scatter them around your garden, it's actually better to plant them in clumps because this will allow bees to remember to come back and know where it is, and there'll be like enough forage. So yeah, natives are really important because native plants are adapted to our climate and our soil. So, and some pollinators are specialists. So these plants are kind of adapted along with our native pollinators. Honeybees, on the other hand, are considered generalist pollinators. They'll visit many different plants, but yeah. And for moths, night blooming, and bats, night blooming flowers. So for butterflies, it's important to include larval host plants. If you want to attract them to your garden, you want to provide them a place to let their caterpillars eat and grow. So yeah, butterflies are considered specialists. So you're going to want to have some milkweeds, and some we have in California are narrow-leafed milkweed, California pipevine, and passion vine. Also have another one. This is another native milkweed, showy milkweed. And if you really want to attract hummingbirds to your garden, everyone knows what color hummingbirds love. What is color? Red, yeah, they love red, but also they love others too. So this hummingbird sage is so beautiful when it blooms. We have it in our haven and the arboretum, but these are just an example of some of the other ones. We also have this on our website. So for bats, less common here to do it, but it's great to point out bats as important pollinators because without bats, we wouldn't have bananas, mangoes, and guavas, to name a few. But yeah, they love the night blooming because they're nocturnal. So thinking about the plants you're choosing, you want to avoid modern hybrid flowers, doubled flowers, because while they're really beautiful and they're dense and they're fluffy, they've taken away the parts that provide pollen and nectar, and they rarely produce seeds. So pollinators will ignore them. And for creating nesting habitat for native pollinators, if you have bare ground in your garden, this allows them, like ground nesting bees, to have a space to live comfortably, but they won't have the strength to dig through mulch. So it would have to be kind of bare. And you can see like C is where they've really flourished. But yeah, this gives them protection. And We also, so you could leave bare nests, but you could also make native bee homes. So let's go to, okay, well, that will come later. So yeah, learn more about pollinators, get some guidebooks, visit the Bee Haven when you get the chance. And if you're really into it, you can really have a blueprint. And when you make your own blueprint for your garden, you want to think about the general size and the textures of your plants, because bees see on the UV spectrum of light, so they see light differently, or the colors differently. So whites, yellows, and purple flowers are really vibrant. So thinking about the shapes of the flowers that will attract the different pollinators is all great. So yeah, this is kind of what they see. So super vibrant. Yeah. So for the native bee homes, this is kind of like what it looks like on the inside. So they lay their egg, and then they get leaves or mud, and then kind of separate that egg, and then keep doing it until the whole tube is filled. This is kind of unlike honeybees. Honeybees, the queen bee will lay all the eggs, and none of the other foragers or worker bees are going to be laying eggs or tending to that brood. But this way, these bees are emerging from this. So you can build your own native bee home. And this is either like you get a wood block and you drill holes into it, or straws, bamboo straws, paper straws are great. And so this is a leaf cutter bee, and you can see it has little yellows carrying pollen in. But if you want to include this in your home, you can, replace them every year, because once they've emerged, they're not going to reuse it again. So you can clean it out with a straw or just replace it entirely. And then you want to be sure to put it in an area that has a solid back, and then like a rain cover or shade cover. Yeah. Yeah, so this is our website, and here you can get so much great information on our plant lists, native plants, and strategies for your own garden. So yeah, thank you so much. And then does anyone have any questions? Yeah.
Not gardening in the soil.
Yeah.
Top 3 or 4 things would you recommend?
Repeat the question.
Yeah, so the question is, if you don't have soil that you're planting in but pots, what plants would you recommend? That is a really great question. So one of the top flowers we have is actually the Mexican sage, and it's this purple flower that attracts every pollinator. And it's super beautiful and vibrant. Like for hummingbirds, hummingbird sage is another one, I would say, because super beautiful flower and attractive. Yeah, rosemary, sage hot lips, yeah, even sunflowers. So, but maybe, yeah. Mhm. Mhm. Any other questions?
I have a quick question. So I read that during the fall,
Uh-huh.
when your leaves kind of drop, you're supposed to actually kind of keep your yard dirty, like
Yeah.
let the debris stay and not put it away. Is that actually accurate? That the bees have
It can-
to overwinter?
It can be, and in like certain pods of certain flowers, that can be an area that they can use as a habitat. So yeah, that can be true. But pruning can also be seasonal. So some plants, we have a pruning schedule that we kind of follow for when things in order to get the best blooms for when they do bloom, we'll prune it on time. But sometimes that is also true. Yeah. Yeah.
I'll follow on to that one.
Yeah.
So when is it safe for breaking the leaves if you're kind of moving them for the winter? Like, are we here now, or do we want to wait till later?
Well, I think you're safe to rake up the leaves, but like in terms of having dead branches or any dead growth on flowers, even though that can be like unsightly, you still are leaving it is not a harmful thing. So yeah, leaves can just wake up. Yeah. Great. And I just want to ask a question to you guys, and I was asking this to our visitors today, but do you know how many native bee species we have in California alone? Or any guesses? Hundreds. It's 1,600 native bee species. So that's why it really makes a difference for everyone to plant their own pollinator gardens, or just even a couple plants in your front or backyard. So yeah, thank you so much.
Samantha, for a really great talk. So we have our next speaker now, and our next speaker is Dr. Tracy Thomson. He's a research assistant and instructor for the Department of Earth and Planetary Sciences at UC Davis, and his research focuses on using modern organisms as models to understand the function and behavior of fossil organisms. He has published papers on Tyrannosaurus feet, fossil reptile tracks, vertebrate claw function, and clamshell structures. And he's currently working on the evolution of semi-aquatic freshwater mammals, invertebrate trace fossils from early Triassic period, and weird dinosaur forelimbs, which he will talk about today. So let's welcome Dr. Tracy Thomson.
Yeah, so today I'm going to be talking about hobnobbing in the Cretaceous. And specifically, I'm focusing on these little dinosaurs, which are called alvarezsaurid dinosaurs, which we'll introduce to you and get used to. Oh, my animations aren't working. That's okay. Okay, so alvarezsauridae, there we go. Forward. They're kind of your standard looking dinosaur. They look a lot like, any other theropod dinosaur. They got the long legs, they're running around. The largest one is only about 2 meters. That's the largest one. Most of them are more like small or medium dog size. So that's interesting that they're not really big as a family group of dinosaurs. But what's also interesting is that they have these little stubby forearms. They're really reduced. Some of them have a single digit, some have 2 digits. Even though they're all really reduced, they all are different. Every alvarezsaurid has a little bit different configuration of its forelimbs, even though they're all kind of stubby like that. So this, of course, prompted paleontologists like, what is that for? Got to get used to the direction that we're going here. Come on. There we go. Forelimb function. What are they using these little forelimbs for? Some people suggested digging burrows or pulling apart termite nests to get at little insects in the ground. Even looking at eggs, maybe holding the eggs. And this is the one that I'm going to talk about today. There was a paper recently published just in December on this new species, which they called Manipulonyx rachetavi. And they quoted saying, these are nocturnal hunters of dinosaur eggs specialized in feeding on eggs. Here's what the forelimb looks like. There's the fossil. You can see it has these little stubby digits. It has these little horns on the palm. That's like right on the inside of the palm. You're kind of looking at the inside of the palm there. And it has this really big digit and claw that comes off here. And they suggest if you look at it in front view that it would hold an egg like this, and this is in side view, and that it could like crack the egg open and eat the egg. If you look at the scale bar on this, that whole forelimb is only about that long. The whole limb is about that long. Right? So if they are eating eggs, they're not really big eggs. And I want to talk about why I think they're not eating eggs and maybe propose an alternative idea for what they might be doing. And this gets me to this concept of Just So stories, which was a Rudyard Kipling production. And this is a collection of children's tales telling the origins of things with fun titles like How the Elephant Got Its Trunk, Leopard Its Spots, How the Alphabet Was Made. These are generally whimsical and fantastic stories. They're untestable narrative explanations. So for example, for the elephant, there's a baby elephant. It's curious about what's going on in the pond. The parents say, don't go down there. It goes down there anyways. An alligator grabs its nose, stretches the nose out. Luckily, the baby elephant can run away and gets away, but it has the reminder of its nose to remind it not to go near the water anymore. And that's how the elephant got its trunk, right? So we as human beings are sometimes really desperate to understand what's going on, that we will even make up or accept explanations that may not be justified, that may be just because stories, right? That aren't really testable in a scientific way. So what I do, and one of the things that I focus on is modern examples, modern animals. And I have a bunch of videos that I would like to have play, but they're not going to play through the thing. Oh, that's a shame. Okay. So this is a snake eating eggs. And it turns out that there's lots of snakes that eat eggs today. There's a species in India, the Indian egg-eating snake. There are 18 species of this genus in Africa that specialize on eating eggs. And these are obligate egg eaters. They don't eat anything else but eggs. And you'll notice that what the snakes are lacking that the alvarezsaurids do is limbs. You don't need limbs to eat eggs, and snakes are the proof of that. Okay? What's really interesting, though, is that's it. There are no other animals in the world that specialize on eating only eggs, only snakes. And only in the tropics, because that's where you don't have seasonality and migrations and stuff, right? There's a constant supply of eggs in the tropics, and you need that constant supply because that's your food source, right? So, what else eats eggs today? Opportunistic oviviry. Mongooses eat eggs, seriemas eat eggs, raccoons eat eggs. Any animal that you give an egg to, it will eat that egg, pretty much. Mongooses, again, I wish I had the video playing. The mongooses, what they'll do is they'll grab the egg and they'll like hike it like a football through their hind limbs against a rock or a wall and break the egg, and then they'll just go eat it. Sometimes they've been known to eat eggs up to 1/3 their own body weight in this method, which is really impressive. But again, no specialized claws, no forearms needed. The seriemas will pick up the egg in its beak and throw it on the ground really hard, and they'll get at the eggs that way. So we don't need special structures or forelimbs to be able to eat eggs. But what do modern animals use their forelimbs to do that are specialized? Well, a couple examples. One is pond turtles, and they will, this is the scientific term for it, it's called titillation. And the males will actually touch and stroke and vibrate around the head of the female. And that's what's kind of being shown here as part of a courtship ritual in mating. As of right now, I've counted at least 26 species. If we look at the males have these elongated forelimbs. You can see them displaying them here. The females don't have that. The males have these really long claws that they use to like touch the female in these ways. And if we include species that we know have those elongated nails that we haven't directly observed this courtship behavior, it's probably closer to 40 species that do this of turtles. And it's species specific. Every species has a very different ritual. Some vibrate, some touch, some come from behind over the top, some come from the front. And they identify each other, and they have this whole ritual that they go through that is species specific. It's only for courtship and mating. Here's another example, cartwheeling in raptors. This is a video of a couple of owls doing it. Here's some eagles. And what these raptors do is they will approach each other, they'll fly upside down, grab each other's talons, and then they'll just cartwheel down like a pinwheel, free falling down to the ground, and then they'll release each other and go off. This has been known to kill some birds. They don't release before the ground, or they hit an object or something like that. And some vultures over in Africa do this from about 1,000 meters up, and they will fall 500 meters or so before actually releasing each other. This seems to have multiple functions. We see males and males engaging in this. We see females and females doing it. We see adults and juveniles, like parent and offspring pairs doing this. So this can be bonding, courtship. It can also be territorial, where you're fighting over land. So there's lots of social functions that raptors use this cartwheeling for. And there were a couple of eagles there cartwheeling down. I don't have the video going for that. So what this brings me to is, like, throughout the animal kingdom, signaling is all over the place. I was talking to a student the other day. It's like us coming up and like shaking hands or smiling at each other, right? There's multiple components, right? There's a ritual to the social interaction. Has to be done in a certain way in order to identify who's good, who's bad, or whatever. And it can be multimodal. There's visual, there's audio, there's touch, there's smell. All of these things are coming together in very specific, complex ways so that animals can identify each other, they can greet each other, they can bond, they can fight, mate, right? All of these social interaction functions. So how did the elephant get its trunk? Well, one of the reasons for the trunk is to touch another member. They will flap their ears at each other. They'll stomp their feet. They'll make grumbling noises. And all of this comes together in this multi-component, multimodal way of signaling back and forth to each other. Here are a couple examples. Grebes do this. They will actually find each other, and then they will pitter-patter and dance across the water in synchronicity with each other. So here we have a very specific, right? The shape, you got to hold your bill the right way. You got to put your wings back. There's the sound of the water churning that's part of this, right? And this is a whole ritual that grebes go through, again, as part of their mating, their courtship rituals. This is a really cool video. This was cordon bleu songbirds. And the male here is holding a little piece of nesting material, which he waves at the female while he chirps, and he jumps up and down on the perch. Which is really cool, and females will do this back at the males too. It's a back and forth thing that they do. This specifically is called the tap dancing bird because when they slow down video of it, what happens is when he jumps off the perch, his little feet do this on the perch really quickly, and you can actually hear it go while he's singing. So again, we have the visual, we have the ritualistic, we have the sound, and all of this coming together in this interaction. And we're just now beginning to understand how complex these interactions really are. For example, this is the bowerbird. We've known for a long time the bowerbird, the male, it builds these structures, it decorates them. The female comes in, he does his little dance, and there's this whole thing going on, right? But what's really interesting is just last month, a paper came out that showed that all these little decorations that he's putting in everywhere actually serve to either amplify or attenuate the frequency, the intensity of the frequency of certain sounds. And so when the female goes in, she'll make chirping noises that will reflect off of different things in the bower, and she's assessing the bower based on that as well as the visual and everything else going on. So very complex, very specific behaviors, social behaviors that these animals are using to find each other and to live their lives. So what about the alvarezsaurid dinosaurs, right? These got these little forelimbs that are really stubby. They're all different shapes and sizes and kind of designs. And I would propose that this may be more for a social function or for touching different members in the right way or whatever, kind of like the turtles, to initiate courtship rituals or to fight with each other or whatever the case may be. And what's really interesting is the case has been made that, well, if you've got this type of sexual selection as mating going on, it's associated with sexual dimorphism, where the males are different than the females, either bigger or smaller. Like the turtles, the males have the claws and the females don't. But in the examples of the birds that I gave, none of that is sexually dimorphic. So we could be seeing dinosaurs and other fossil organisms that are not sexually dimorphic, that all have crests and horns and little protrusions and things. And just because we're not seeing the sexual dimorphism doesn't mean that there's not this incredible complex interaction going on, utilizing those structures, those weird structures, to initiate and send those signals and to receive them amongst pairs. This is a Diplocaulus that has this famous boomerang-shaped head, and lots of ink has been spilled on, oh, is that a hydrofoil? Is it helping it, in a stream or to stay on the bottom or whatever? Well, what if it's just like the turtles that are going around and touching each other and stroking along the sides or something? I don't know, right? But I think this is because we're looking at this in the modern world, and we're just now starting to see how complex and pervasive the signaling is. I think it's something that we need to consider for the fossil realm as well, just as equal. And as has been said also today, collections are extremely important. I wouldn't be able to do what I do without collections. Without living collections, in the sense of they are there, they're available for study, I can go see them, anyone can go see them. And not just collections of objects, but books and obviously specimens in museums, but also zoos. And like our Raptor Center here, you can go see these live animals touching each other, interacting with each other, how they're responding to each other, right? These are living, breathing animals. And for a paleontologist like me who studies dead things, the living world is crucial to understand the dead things as well. So with that, I'll finish there, and if there's any questions, yes?
So that's Mononicus, right, on the picture?
I don't know if this is Mononicus or not. It might be. I think it
I
is.
was gonna ask, because it's often like shown as like barn owl.
Yep.
So is there like really, is that like a creative choice, or is that like what we know it like?
Pretty much 90% of what you see on a TV screen is creative choice. So with this little guy, Mononicus, yeah, it's usually shown like going and tearing into wood, like termites, eating termites or something like that, right? But my argument is that, this is a close quarters thing. Things that tear into termites today have much longer limbs. If you're defending yourself against a predator, you want to keep them at a distance. You don't want them right here, right? But what if it's your own species? What if it's a turtle that you're trying to interact with and get some social commitment? Well, then that's up close, that's personal. And so you would bring them in close to be able to do that thing. There is some evidence that the alvarezsaurids were nocturnal, and being really small, you're running around in the jungle, it's dark, there's lots of obstructions. You can use sound to find each other. Once you've found each other, you don't want other things finding you, so you switch modalities to something like up close touching, cooing, vibrating, or something like that. And we see that in the animal kingdom too, where they switch modalities depending on distance, depending on the medium and that sort of stuff. Yeah. Okay?
I had a question.
Yes.
So a lot of these are shown, it looks like they have feathers.
Yes.
So is there support for that?
Yep.
They would have?
Yeah, this is a group of dinosaurs that shoots off just before the raptors and the birds. So they're very closely related to the bird clade. They're up in that part of the dinosaur family tree. Yeah, and so we have evidence of feathers. And so again, you're talking about like not just the tactile, but there's visual components, colors, sizes, and shapes of feathers could all be part of that as well. Yeah. One more? Sorry. Go ahead real quick. Yeah.
In the debate with this, it actually sounds really similar to the way that we talk about why T-Rex have the little arms and why Predators have little arms. Is there like any connection between those, or is that just like a coincidence?
Maybe. So that's why I said, like, when I was doing the research on this month, I'm preparing the paper on this, it's surprising to me just how recent we've realized how complex this signaling is and the role that it plays in evolutionary biology and sexual selection and all of that. So I think it's too new, and I don't think a lot of paleontologists have really grappled that this could be just as pervasive in dinosaurs or other fossils as in the modern world, and why wouldn't it be? Yeah.
Thank you for the talk, Tracy. It was really, really interesting. So now we talked about nematodes, we talked about bees. So the next stop is Dr. Kyria Boundy-Mills, and she has been the curator of Phaff Yeast Culture and Collection in the Food Science and Technology Lab since 2001. And her outside interests include genealogy and playing keyboard with the jazz big band. And today, she will talk about the many ways that yeast from the Phaff collection are being used to solve problems and make discoveries. So let's give her a big hand.
Can you hear me okay? Yeah, great. So thank you for the introduction. I am the curator of the Phaff Yeast Culture Collection here at UC Davis. And thank you for coming to Biodiversity Museum Day and showing support for collections. So first of all, what are yeasts? Yeasts are a living organism. They are single-celled fungi. They're fungi. They're not plants. They don't have chloroplasts, so they can't do photosynthesis. They rely on eating sugars from their environment. In other words, they eat dead stuff. They're degraders. You would find them in large numbers where you would have moisture and a carbon source like sugar, for example, in fruits, in the nectar of flowers, in tree saps. You would also find them where there's any degrading organic material, dead animals, dead plants. A few can live in living organisms. A small number of yeasts can cause infections, but that's just a few of their yeasts. There's about 2,500 species of yeast that have scientific names, but only about 5% of yeast species have been brought into the lab and given a scientific name by scientists. So 95% of yeasts are out there in the environment waiting to be named. They are hard to identify the way plants and animals are by the shape. I've shown a few shapes here. They're little round blobs, little oval blobs, and that's about it. Sometimes they stay together in small chains, but there's not much diversity in the shape of them. So when we use DNA now for identifying these species. One example of a yeast species is Saccharomyces cerevisiae. This yeast in nature is found in very few places. You've heard about specialists among the different animals and plants today. Some yeasts are also specialists. This yeast has only been found in nature in high sugar fruits, just a few fruits, and the insects that visit them. They're found in smaller numbers, like in tree sap, in the soil below a tree that's dripping sap, and that's about it. It eats sugar, of course, like yeast do. It produces carbon dioxide and ethanol after it eats sugar. That's the carbon dioxide is the bubbles in your bread, or as I like to call them, yeast farts. And they also produce ethanol. The alcohol in your beer, the ethanol that we use for our cars, is produced by this species. So this species is used to make wine, beer, bread, other fermented foods and beverages. It's used to make biofuels, chemicals. It's also been genetically modified to make other very useful products like insulin, growth factors, antigens for vaccines, and other important products. It's also used for basic science discoveries. Since the year 2000, there have been 5 Nobel Prizes for major discoveries using yeast as a model organism. You have 20,000 genes in your genome. Yeast has a very similar version of 26% of those genes. Genes for basic functions like copying DNA, making proteins. So we can study those processes that happen in our bodies by using yeast. I am the curator of this very large yeast collection. It's called the Phaff Yeast Collection in honor of Hermann Pfaff. He spent his career studying yeast ecology and taxonomy. He had over 300 scientific publications, which is a lot. And for his research, he collected yeast from 50 countries and built a collection of over 6,000 strains. So he discovered the basic concepts of yeast ecology. And some of the concepts are that some yeasts are generalists. They can be found in many different places. Some yeast species are specialists, like Saccharomyces cerevisiae. It's only found in a few types of locations. Some yeasts can interact with other organisms in their environment, such as insects or plants. They cooperate with some organisms and compete with others. For example, in this photo, this is Hermann Pfaff. This is Hermann Pfaff collecting yeast from rotting cactus. He has a Petri plate in one hand and a sterile loop in the other. So he would scoop up the rotting cactus and streak it on an agar plate, and the yeast would grow. So he was studying why certain yeasts are only found on certain species of cactus, and how they interact with, in this case, with the Drosophila fruit flies that lay their eggs and can breed in rotting cactus. So in this study, which spanned many years, he discovered that yeasts provide a source of protein for these fruit fly larvae, and yeast and the flies, to return the favor, flies transport yeast to the next rotting cactus, because yeast don't have wings, they don't have legs, so they rely on a vector to get to the next cactus. All right, and also yeasts compete with other yeasts. Some yeasts produce a protein called a killer factor that can kill some of their competing yeasts so that they can dominate in that habitat. So yeah, I just summarized about 20 years of research, but in the process of making these really fundamental discoveries, he built this huge collection. He named 50 yeast species. There were 700 species that had names when he died. He named 50 of them. All right, and built this collection up to 6,000 strains. I worked with Hermann Pfaff for 2 years. He was still coming to work every day till he was 88 years old. And luckily, I overlapped with him for 2 years. So when he died, I was in place to take over the collection. And I have expanded it to over 10,000 strains. We have about 1,500 species in the collection. And I have rescued the collections from 3 other professors at other universities when they retired. I use this collection to help other researchers. I distribute yeast to people at other universities, helping them with their research, to companies, to government agencies that are doing yeast research. And I help them to select yeasts that are the best fit for their research project based on my knowledge of yeast, my knowledge of the collection, and a huge amount of unpublished phenotype data that was used to identify the species. And I also perform research that builds and uses the collection. This is the 4th largest collection of its kind in the world. There are very few places where we can do this kind of research, pulling out hundreds of species and comparing them. So just a few things that I've been researching the last several years. One is high oil yeasts. So most yeasts will eat sugar until the sugar is gone in their environment, and then they stop growing. There are a few yeast species that will continue to eat the sugar, and they will store it inside the cell. So what this is one yeast cell that we've stained with a fluorescent dye. And these glowing balls inside the yeast cell are big lipid bodies. These yeasts can be 50% oil. Put that in perspective, a soybean is 18% oil. That's one of our major sources of vegetable oil. These yeasts have way more oil in them. And different yeasts have different kinds of oils. Some are liquid and some are solid. So there's a lot of variation in these types of oils. And so we're trying to find oils that meet specific needs, such as replacing animal fats in a meat substitute, replacing tropical oils like palm oil. All right. So, and there's another area of research. We were growing some of these high oil yeasts, and the people in my lab, Irna, Antonio, noticed that there were these little droplets outside of the yeast cells. You could see it with your naked eye. And when the culture grew a lot, we centrifuged it down, and the oil went to the bottom of the tube. Oil is supposed to float. So we knew that it was a new thing. And it turns out that this material that the yeasts were naturally secreting it's half sugar and half oil in one molecule. And so it's a glycolipid, and it has a natural detergent property. So a company licensed our technology, licensed our yeast strains, and within the next year, there will be consumer products that you can buy that have this new natural detergent in it. So stay tuned. Okay. Another project I was involved in several years ago, an olive company was having problems with spoilage. So this is an olive that's spoiled. It's all squishy and soft. We isolated hundreds of yeast and were able to recreate this softening by adding specific yeast to a bucket of raw olives. The olive that caused the spoilage was a Saccharomyces cerevisiae. Okay. All right, so I'm going to tell you more about the collection and how a yeast that was originally isolated for one purpose ends up being used for other purposes. All right, so here's our olive spoilage yeast just growing on a plate. And in 2011, I was contacted by a group in France. They wanted to sequence the whole genome of 1,000 separate isolates of Saccharomyces cerevisiae. We sent them a few strains. They got strains from collections and labs all over the world. They published it in Nature, which is a really good journal. And so this is kind of a figure showing how different strains of Saccharomyces changed over time and in different parts of the world. So it was a great paper. They made these 1,000 genomes publicly available in a database. In 2020, a biotech company mined that data, and they were looking for a yeast that made a specific enzyme called asparaginase. So why asparaginase? So asparagine is a protein that's asparagine is an amino acid that's naturally found in proteins in our food. If you heat that up, if you fry that material, some of the asparagine turns into acrylamide, which is a toxic compound. You don't want too much acrylamide in your food. They use this yeast that it causes spoilage, but it also produces asparaginase. They improved it, and now that yeast is available to food producers to help reduce acrylamide in the foods that we eat. So the villain that was causing spoilage in the beginning became the hero that is making our food safer. The yeast is also used by researchers around the world. We just sent 60 strains to a group in Slovenia that is, they got 200 strains total of this yeast called Hanseníaspora. That's a fruit-associated yeast. And they sequenced the whole genomes, and they're showing how the genome changed over time as the yeast got migrated to different parts of the world. It's a really cool story. And there's a group at USDA is using our yeast to produce, try and control spoilage on produce. So these yeasts are being used in so many different ways by lots of people around the world. And of course, I don't do all this work myself. I rely on my lab, including my lab manager, Karen. So I would be happy to answer a couple of quick questions. And so thank you for coming today. Yes, sir. What is my favorite kind of yeast? Don't make me choose. Oh, you'll break my heart. That is one of my two kids. Don't make me choose. So I like Saccharomyces cerevisiae because it's so well used. It's used for so many different purposes. Part of my goal is to show that there's so many other species that could also be used to make foods, to make medicines, to make chemicals, to make basic scientific discoveries. So I'm helping to get all the other yeast species to have some publicity like Saccharomyces does. Yes, what's your question? The tastiest. So we are developing these yeasts to make new food ingredients. There's a small number of yeast species that have been given approval by the FDA to use them as a food ingredient. Saccharomyces, of course, for beer, bread, and wine, and a few others. The yeasts that we're using to make oils, they are not on that list. They're totally experimental. So I can't taste them, but I can tell you, when we were extracting the oil from some of our yeasts, we put it in this little press in the lab, and it was pressing the yeast really hard with some heat. The lab smelled like frying chicken. So I couldn't taste it, but I happened to smell that yeast was really delicious smelling. Yes?
So you go out into nature and you swab a cactus and you put that on a petri dish.
Mm-hmm.
I'm assuming that you swab, you get more than just one species of yeast. I'm assuming you get multiple kinds of yeast, bacteria, mold. How do you then look at what's on your petri dish and isolate individual kinds?
That's a good, yeah. So if you just take a plant rot and spread it on a plate, you'd have to spread it pretty thin because the initial glop of plant rot would have way too many things to get in individual colonies. So we spread it, and then we spread it thinner, This is called streaking for single colonies. So by the end of spreading it out really thin, you would have individual cells land on that plate. And then each individual cell divides and divides and divides to have 1 million cells on that spot. That's when you can see it with your naked eye. So I don't have them on this talk, but on the original isolation plate, you would have different, they're called colonies. Different colonies are slightly different shapes, different colors. Most of them are kind of creamish to white. So what we do to make them look even more different, we use a special dye. It's called rose bengal. It's this beautiful magenta colored dye, and different yeasts will pull up that dye to different degrees. So some of the colonies will be this beautiful red, dark pink. Some will just be really, really faint pink. So things that look almost identical on a regular plate can have more color variation. Yes?
So when you isolate a strain, how do you ensure that it will always be the same strain? Because every time they divide, they differ from the first sample, right?
Right. So every time you streak it onto a fresh plate, there's going to be many generations to form a whole colony. So that's called genetic drift. So we try to reduce the number of times that we actually grow it and then freeze it again, grow and freeze it again. Before my time, Herman Pfaff kept his collection in a very inexpensive but very labor-intensive method, and that was on auger slants. So every 5 years, it had to be put on a fresh slant. And some of those had been going through that process every 5 years for 50, 60, 70 years. So there's probably some genetic drift. So one of the first things I did when I took over the collection in 2001, I got some money from the campus to get everything out of those auger slants and into vials in the freezer. Because once they're frozen, I have a -80 Celsius freezer. Once it's frozen, it will just stay dormant and not do any more genetic drifting. So that's one of the first things I did. Yes.
So of the yeast that produce oil, are there opportunity for industrial sort of applications of that to replace fossil fuel or other things that we use?
Yeah, so these oils that these yeast produce, they're similar to plant oils, like vegetable oils, the triacylglycerols, C16, C18. So they're similar to like vegetable oils. They could be used to make biodiesel. They could be used, technically, theoretically. The problem is it costs more to make that kind of oil than it does to make a plant oil. So so there's a couple of strategies to make that technology more competitive. One is reducing the input costs, reduce the cost of sugars that you feed to it. We're trying a whole bunch of different waste streams, almond hulls, leaves, grape pomace, tomato pomace, things like that. So reducing this cost. Another strategy is it's actually what's used in the petroleum industry, the soybean oil industry. You don't just sell the oil. You sell like for petroleum, you market the oil, the kerosene, the diesel, you fractionate it and sell all of those parts. So we are looking for a yeast that can make the oil plus protein, plus vitamins, plus a pigment, plus carbohydrates from the cell wall. So we're looking for the ideal yeast that has all of those parts that we can separate and sell. Yeah, yes.
So I was just wondering, you have so many yeast in the collection. Like, is there like a database people could visit to see like what kind of yeast you have and like, what they can do and stuff like that?
Yeah, so the question was, do we have a database that people can look at? Yeah, and the answer is yes. We have a website with an online catalog. It has 10,000 strains in it. You can search our online catalog by genus, species, where and what part of the world it's from, what kind of habitat it's from, whether it's from food or animals or, and so that's at pfaffcollection.ucdavis.edu. And for researchers, you can put the yeast in your cart and you can send me an order and we will prepare a little slant and ship it to you for $300 a strain. Yeah. It's a lower price for collaborators. I send them to collaborators for free. All right, so I went way over time, so I'd be happy to answer any questions individually. So thank you all for coming for inaugural speaker series. It was really fun. It was a very fun day. Thank you all for coming.